This is me Arzu here and I welcome you to my channel Mr. Teeth. We get dental concept get dissected. So this is our part 3 video of the alveolar bone, and in this video we will be discussing with the intramembranous ossification, endochondral ossification, and a small topic that is Orban bone versus lamellar bone. Okay, so in this video we will be studying in detail about the ossifications. Okay, so all those notes are prepared from Orbans. Okay, so I'll be dealing properly with the every lines of Orban. Okay, and I think after this completion of this video you will be mastered in this topic, intramembranous ossification and endochondral ossification. Okay, and also those topics are sometimes asked in exams, okay, in a short note, but it is not much important for the oral histology, but also once you should look this topic because it is a kind of a general topic. Okay, so now without delaying much, let's start our today's video. So our first topic is intramembranous ossification. In intramembranous ossification, okay, so the main thing you should understand is that, okay, so direct formation of the bone within a highly vascular sheets of the condensed primitive mesenchyme. Okay, so there is an intramembranous and another is a endochondral. Endochondral means there is a replacement of the cartilage to the bone. Okay, so means cartilage is changed to the bone, there is a proper replacement of the cartilage into the bone. But in intramembranous ossification, okay, so there is a condensed primitive mesenchyme. Okay, so that change into the bone. Okay, so that is the main difference between both in ossifications. After that it occurs in the flat bones of skull and the clavicles. Okay, so this is also very important. After that it begins toward the end of the second month of the gestation. Okay, it begins at the end of the second month of the gestation. Okay, so this is an introduction to the intramembranous ossification and this is important. Okay, so just remember it is formed from the vascular sheet of the primitive condensed mesenchyme. That is important here. After that it occurs in a flat bones like skull and clavicle. Okay, so these are the flat bones. And about the classifications of the bone, okay, so I have discussed that in a first video of the alveolar bone. Okay, and this date at the end of the second month of the gestation, so this is also very important. After that now we will discuss with the sequence. Okay, so in which sequence, so there is a proper sequence in which there is a intramembranous ossification taking place. Okay, so our first step is a formation of the bone matrix within a fibrous membrane. Okay, so there is a formation of the bone matrix within a fibrous membrane. So in the first point, as there is a, okay, so there is a loose mesenchyme containing the stellate cell, okay, with interconnecting cytoplasmic process. Okay, so about this stellate cells, okay, so these stellate cells are quite of similar that of the enamel organ. Okay, so in development of the teeth we have discussed with the stellate cell. Okay, so it is like this. Okay, so this is a stellate cell. Okay, another stellate cell is like this. Okay, and they are connected by the cytoplasmic processes. Okay, so they are connected like this. Okay, so they are connected with each other, so there is a proper network between the stellate cells. Okay, so those stellate cells are quite of star like. Okay, and they have interconnecting cytoplasmic processes and they present in this loose mesenchyme. Okay, after that center of osteogenesis develop in the association with the capillaries. Okay, so this is very important. Okay, so this is the main inducing point of this ossification. So center of osteogenesis. Okay, so center of the osteogenesis is developing but in the association with capillaries. Okay, so that is very important. There is a association of the capillaries, there is a association of the blood vessel. Okay, in this ossification. After that mesenchymal cells in the center become round and basophilic. Okay, so at the center these mesenchymal cells, okay, at the center become round and basophilic, and after that those mesenchymal cells secretes the organic bone matrix and the matrix soon begins to calcify. Okay, so just in this first step of the intramembranous ossification, okay, so there is a condensed mesenchyme. Okay, condensed primitive mesenchyme in which there is a stellate cells which have interconnecting cytoplasmic processes, and in those loose mesenchyme, okay, so there develop a proper capillaries. Okay, so there is developing of the vascularization. So due to the vascularization, there is an induction of the ossification, and the mesenchymal cells that is just adjacent to those capillaries are secreting the organic bone matrix, and those matrix becomes calcified, okay, soon. And about this, okay, so mesenchymal cells show round and basophilic type of properties when they are just adjacent to the capillaries and when there is an induction of the ossification, okay. So this first step is very easy to understand. After that, in a second step, there is a formation of the Orban bone, okay. And about the Orban bone and the lamellar bone we have discussed properly in the first video. So this Orban bone is a immature bone, okay. After that, in first point here, so first, small newly formed bone matrix is irregularly shaped spicule, okay, and this spicule term is important. So first, newly formed bone matrix, okay, not bone matrix. In this bone matrix we will deposit the mineral and there will be proper bone. So this is just a bone matrix. So first, small newly formed bone matrix is irregularly shaped spicule. After that, spicule lengthens gradually and anastomosing forms the trabeculae, and those spicules, okay, so they lengthen, so they just grow in length, okay, and they just interconnect with other spicules too, and they form a network of spicules that is called trabeculae. And trabeculae extends in a radial pattern leading to the network of the trabeculae which is present in a spongy bone, okay. So this sequence you should learn, okay. So formation of the Orban bone. Formation of the Orban bone means, first of all, there is a formation of the spicule, okay, and spicule is a bone matrix, irregularly shaped spicule means bone matrix, okay, and those bone matrix they just gradually lengthen and they anastomose with each other, okay, and form the trabeculae, and further this trabeculae again grow in a radial direction and again they network with each other trabeculae and they form the spongy bone, okay. And in the spongy bone we have different trabeculae there, okay, so we have discussed with this. And after that, few undifferentiated mesenchymal cells leave a layer of the flat cells called osteogenic cell on the trabeculae, okay. And if there is a rich vascular area, then it forms the osteoblast, and less vascular area, okay, less vascular supply is there, then it forms the chondroblast, okay. So formation of the Orban bone. So in this step there is a formation of the bone matrix, or you can say immature bone, okay. So there is a, first of all, formation of the spicule, okay, which is irregularly shaped and it is newly formed bone matrix, and those spicule they just connect with another spicules and they form the trabeculae, and those trabeculae grows in the radial direction forming the network, okay, and those network is called spongy bone, okay. So those networks of the trabeculae form the spongy bone, and the few of the undifferentiated mesenchymal cells, okay, so those mesenchymal cells are going to be changed into osteoblast, okay. So those undifferentiated mesenchymal cells, if there is a rich vascular supply, then it changes to the osteoblast, and if there is a less vascular supply, then it changes into the chondroblast, okay. So this is very important to understand, okay. After that, in our third step, so we have appositional growth mechanism and formation of the compact bone plates, okay. So oppositional growth, we have discussed with this apposition in the development of the teeth, okay. So it comes under the physiological stages of the tooth development, so we have discussed there. So similar here, appositional growth mechanism, okay. So new layer of a bone matrix are deposited on the pre-existing bone surface, okay. So the minerals are deposited in the layers, okay. So they are deposited in the layers, okay. So one layer, after that another layer, after that another layer, okay. So we can see the layers there. After that, osteogenic cells are always on the superficial position, repeating the process again and again, okay. So if this is a, just think this is a first surface of the bone, okay. So we have cells here, okay. So I will draw with another pen. So osteogenic cells are there, okay. So this is osteogenic cells, okay. We have osteogenic cells, okay. So these are osteogenic cells, okay. So they are always on superficial position, okay. If upon this, okay, another layer is if upon this another layer of mineral is deposited, okay, then those osteogenic cells, okay, those osteogenic cells, okay, they at the superficial position they just differentiate into the osteoblast, and after that osteoblast they become the osteocyte, okay. So I will be telling that later on. Okay, so osteoblast and osteocyte. Okay, so I have discussed about this in a previous video too. Okay, so osteoblast comes and change into the osteocyte. Okay, so those superficial osteogenic cells, okay, they change into osteoblast, and when the mineral is deposited upon them, okay, so they just get entrapped into the bone, okay, and they form the osteocyte. So that is the concept. Okay, so osteogenic cells are always there, okay, on the surface. Okay, they just secrete the bone matrix, and upon which mineralization takes place. Okay, so they are responsible for those. And osteogenic cells means it is differentiating into the osteoblast. Okay, so it is quite a similar thing. So osteogenic cells are always on the superficial position. After that, continued oppositional growth and remodeling of the trabeculae converts the cancellous bone to the compact bone. Okay, so continuous oppositional growth is there, and also there is a remodeling too. Okay, so sometime during the mineral formation, okay, in the bone, so we need remodeling too, okay, to give a proper shape and structure. Okay, so that is here remodeling of the trabeculae converts the cancellous bone to the compact bone. Okay, our fourth step is the formation of the osteon. Okay, and osteon means the Haversian system. Okay, so formation of the osteon. So we have discussed with the altogether three steps, that is a formation of the bone matrix within a fibrous membrane. Okay, so first of all there is a formation of the bone matrix, and upon those bone matrix there is a formation of the Orban bone. Okay, immature bone is there formed. After that there is oppositional growth, okay, and remodeling of the trabeculae to convert into the compact bone, and at the last we have the proper Haversian system formation. Okay, so after the bone tissue build up by the opposition, trabeculae thickens and the soft tissue space get narrowed. Okay, so when the bone tissue, okay, is built up, okay, so when there is a proper completion of the apposition, and after that trabeculae, okay, so trabeculae are thickened, so they are thickened and the soft tissue get narrowed. Okay, so trabeculae are thickened and the soft tissue get narrowed, and after conversion to the compact bone, numbers of the marrow canals are formed lined by the osteogenic cell. This is important here, and those canal encloses the vessels and give rise to the system, that is Haversian system or the osteon. The osteon is a primitive osteon. Okay, okay, so this formation of the osteon, I hope you know about this osteon, Haversian canal. Okay, so I have discussed properly in the bone histology. So in this step, okay, of the osteon, we can see the proper development of the vascularization inside the bone. Okay, so in the Haversian canal there is a running of the blood vessels. Okay, so the blood vessels are enclosed on those canal. Okay, so we can see the proper vascularization of the bone in this step, of the Haversian system formation. Okay, and this trabeculae thicken here, okay, and those thickening of the trabeculae, okay, and the soft tissue get narrowed. Okay, so soft tissue it becomes narrow there, soft tissue it decreases there, and we can see different kind of canals formation in the bone, and within those canals we can see the blood vessel running in it. So that forms the Haversian system. Okay, so we have completed with this intramembranous ossification, and this is very easy to understand. Okay, and one note is here, BMP bone morphogenetic proteins and transcriptional factor CBFA1 in the intramembranous ossification. Okay, so they are important chemicals. Okay, they are important transcriptional factor or the proteins important in this intramembranous ossification. Okay, this can be framed into the MCQ question. Okay, after that we will discuss with this endochondral ossification, and this is also very important. So ossification involves the replacement of the cartilaginous model. Okay, so from the cartilage there is a formation of the bone, endochondral ossification. Okay, it occurs at the extremity of the long bones, vertebrae, ribs, articular extremity of the mandible, and the base of the skull. Okay, so endochondral ossification it occurs at the extremities, long bones extremities, okay, and vertebrae and ribs, okay, articular extremities of the mandible and the base of the skull. So those are the places. After that we will also discuss this endochondral ossification in the sequence, okay, as we have discussed like that of the intramembranous ossification. So here also in the first step we can see the formation of the cartilaginous model. Okay, so here is the formation of the cartilaginous model in the first step. So condensation of the mesenchymal cell, which is similar with that of the previous, and differentiate into the chondroblast and form the cartilage matrix. Okay, so here first of all there is a differentiation into the chondroblast itself. Okay, but there, okay, the osteogenic cell may differentiate to the osteoblast or the chondroblast based on the vascular supply, as we have discussed here. Okay, so don't get confused. So here this vascular area osteoblast, less vascular area chondroblast, but here the mesenchymal cells, they differentiate into the chondroblast and form the cartilaginous matrix, result in the development of the hyaline cartilage model. Okay, so cartilaginous model is formed there. Okay, hyaline cartilage model. Okay, so this is also very important. Which cartilage model? We have different cartilages. Okay, so this hyaline cartilage model, and after that it begins late in a second month of the development, which is similar with that of the intramembranous ossification. After that, in cartilage. Okay, so if you have studied the histology of cartilage, the outer layer of the cartilage is called perichondrium. Okay, and we have outer fibrous in that also, and also inner cellular or chondrogenic layer. Okay, so in perichondrium itself we have two different layers, outer and inner. Outer is fibrous, inner is a cellular or the chondrogenic layer. After that, there is a growth of the cartilage model by the interstitial and the appositional growth. Okay, so cartilaginous model is increased in a size by the growth. Okay, and that is by the interstitial and the appositional growth. So increase in length by the interstitial growth, and widening of the model is due to further addition of the matrix to its periphery by the new chondroblast, and that is a appositional growth. Okay, so in this point, okay, so in this step just okay, so there are a lot more points, but first of all just learn this much. Okay, so the main concept that you should take from this, okay, this cartilaginous model formation. Okay, so how it is formed is that, so first of all there is a condensation of the mesenchymal cells, and those mesenchymal cells are changed into the chondroblast, but it is not similar in the intramembranous. It may differentiate into the osteoblast or the chondroblast based on the vascular supply, but here mesenchymal cell first of all change into the chondroblast, and those chondroblasts secretes the cartilaginous matrix. Okay, and that is a hyaline cartilaginous model is formed there. It begins in the second month of the development, and the outer covering of the cartilage we have perichondrium. In perichondrium also we have two different layers, outer fibrous and inner chondrogenic or cellular. After that, and another point, okay, so we can see the growth of the cartilaginous model. Okay, so there is a growth of the cartilaginous model, and that is by the two way. One is appositional or another is interstitial. Okay, so interstitial growth helps in the increasing in the length of the cartilaginous model, and this appositional growth, okay, so matrix are added at the periphery. Okay, so if this is a small circle, then matrix are added at the periphery. Okay, so new matrix are formed at the periphery, so that this circle become larger. Okay, so it becomes more larger, it becomes now more bigger. Okay, like this. Okay, so there is a widening of the model. Okay, that is by the appositional growth. So width is increased by the appositional growth, length is increased by the interstitial growth. Okay, and interstitial growth means between the two structure, it's called the interstitium, and in the at the interstitium there is a deposition. Okay, so that is an interstitial growth. Okay, so just think interstitial length, appositional, widening. Okay, okay, after that cell organized into the longitudinal columns, and that are divided into the three zones, and this is very important. Okay, so in the short note they used to ask this. Okay, write the three zones, okay, that is taking place in the endochondral ossification. So the cells, the mesenchymal cells, okay, they organize into the longitudinal columns, and they are subdivided into the three zones are there. So zone of the proliferation, zone of hypertrophy and maturation, and zone of the provisional mineralization. Okay, so the zone of the proliferation means cells are smart and cells are small and flat. Okay, so sorry, smart is written here. Cells are small and flat and constitute of the source of the new cells. Okay, so they are small and the flat and constitute the source of the new cells. Okay, so that is the zone of the proliferation. Okay, and at this zone of the proliferation we can see the proliferation of the cell. Okay, so we can see the divisions of the cell, and the cells are small and flat there. Okay, after that another zone is a zone of hypertrophy and the maturation. So it is the broadest zone. Okay, so zone of hypertrophy is the broadest zone, and in this zone chondrocytes, okay, so chondroblast forms the chondrocyte as the osteoblast form the osteocyte. So chondrocytes hypertrophy is here, it secretes the type 2 collagen, okay, in the early stages, and later secretes the proteoglycans. Okay, so chondrocyte it secretes the type 2 collagen in the early stage, but in the later stage it forms the proteoglycan, and when the chondrocyte reaches the maximum size, it secretes the collagen type 10 and non-collagenous protein. Okay, and those type 10 collagen and non-collagenous protein, they partially break down the proteoglycan, and this forms the matrix environment receptive for the mineral deposition. Okay, so this is a proper mechanism, you can learn this. Okay, so at the zone of proliferation, there is simply the proliferation of the cells, and cells are small and flat there. And in the zone of hypertrophy and the maturation, there is a enlargement of the chondrocyte, so it is called hypertrophy. And maturation means, okay, it forms the receptive environment, okay, for the mineral deposition. Okay, so there is a maturation of the bone, so that is why it is called maturing zone. Okay, so it is broadest chondrocyte hypertrophy is there, type 2 collagen, proteoglycan, type 10 non-collagenous protein secretion is there, and that is causing the breakdown of the proteoglycan which was secreted earlierly, and this forms the receptive for the mineral deposition. Okay, so it is very simple to understand if you remember this. After that, we have zone of the provisional mineralization, so matrix mineralization by the formation of the matrix vesicle. So in this zone there is a mineralization, okay, by the matrix vesicle. Okay, so first zone, it is a zone of proliferation, second zone is a zone of hypertrophy and the maturation, and our third zone is a zone of provisional mineralization. So now in this step of this second step of this endochondral ossification, okay, so you should learn this, that capillaries grows into the perichondrium. Okay, so this blue, okay, so that I have made here. Okay, so this blue dots are the capillaries, okay, and this green is a perichondrium. Okay, so in the mid section of the bone, okay, so we can see these capillaries are growing inside the perichondrium. Okay, so it grows towards the perichondrium, okay, and it surrounds the perichondrium, and the cells, okay, that are just inside this perichondrium, okay, those inner cells differentiate into the osteoblast in the presence of those capillary and the vascularization, and they form the osteoblast and they form the bony collar. Okay, so bony collar is deposited inside this perichondrium. Okay, so that is the proper method of the formation of the bony collar. After that, we can see the formation of the periosteal bud. Okay, so periosteal capillaries accompanied by the osteogenic cells. Okay, so periosteal capillaries, so periosteal means at the periosteum. Okay, so capillaries at the periosteum, as I have written here at this stage, perichondrium. Okay, so this is a perichondrium, green color, but with the development of this capillaries and the proper vascularization and the proper development of the bony collar, the perichondrium is named, referred as a periosteum. Okay, so the capillaries that is associated with the periosteum are the periosteal capillaries, and they are accompanied with the osteogenic cells, embed the calcified cartilage in the middle of the model. Okay, so this is the periosteal bud. Okay, so this is a periosteal bud. Okay, so this periosteal bud contains the periosteal capillaries and the osteogenic cells. Okay, so that is a two main constituent of the periosteal bud. Okay, so as I have written here, periosteal capillaries plus osteogenic cells, they form the periosteal bud, and they act as a primary ossification center. Okay, so this is important. Okay, so it acts as the primary ossification center, this periosteal bud, that is, okay, that is formed by the periosteal capillaries and the osteogenic cells, and they finally invade into the calcified cartilage in the middle of the model. Okay, so they are invading this calcified cartilage. Calcified cartilage means the bony collar. Okay, so it is referring to that bony collar. So woven bone type structure. Okay, immature bone is there. Immature bone means that is little calcified cartilage type. Okay, okay, after that, osteogenic cells give rise to the osteoblast, deposit the bone matrix on the residual calcified cartilage. Okay, so osteogenic cells give rise to the osteoblast. It is sure that osteogenic cells will differentiate into the osteoblast, and they deposit the bone matrix on the residual calcified cartilage or that bony collar, and that is called mixed spicule. Okay, so as a spicule was just the bony matrix, okay, that we have discussed in the intramembranous ossification, but this mixed spicule means we can see bone also there, means some calcified tissue also there, and also the bone matrix there. Okay, so that is a mixed spicule, and network of the mixed spicule is called the primary spongiosa. Okay, so this can be asked in your MCQ exam. Okay, so this is very easy to remember. Okay, so first of all, there is a formation of the, as in this paper, okay, as I have taught you. So this is a formation of the cartilaginous model. Okay, so there is a proper cartilaginous model is formed there, and we have discussed with the three zones there, and there is a formation of the bony collar. After the formation of the bony collar, we refer perichondrium as a periosteum, and there is a formation of the periosteal boards, and those periosteal boards contain periosteal capillaries and the osteogenic cells, and these osteogenic cells differentiate into the osteoblast, and those osteoblasts deposit the bone matrix on the calcified cartilage, invades into this calcified cartilage and deposit the bone matrix there. Okay, so they form the mixed spicule, and the network of the mixed spicule is a primary spongiosa. Okay, so this much is clear. Now after this, in a fourth step, so there is a formation of the medullary cavity. So about this medullary cavity, so this is a present in the bone, okay, in the center of the bone. So it is the primary ossification center. Okay, so this is a primary ossification center. Okay, so periosteal board forms the primary ossification center. Okay, so this primary ossification center, it enlarges and is spreading proximally and distally. Osteoclast breaks down the newly formed spongy bone. Okay, so this primary ossification center, it just enlarge and spread, okay, proximally and distally too. Okay, and the osteoclast, that is the bone resorpting cell. Okay, so they just break down the newly formed spongy bone, and this breaking of the spongy bone, okay, newly formed spongy bone breakdown forms the medullary cavity in the center of the shaft. Okay, so this medullary cavity is formed in the center of the shaft or the diaphysis, and after that hematopoietic stem cells enter the medullary cavity, giving rise to the myeloid tissue. Okay, so this myeloid tissue are responsible for the blood cells formation. Okay, so they are stem cells. Okay, myeloid tissue, so their stem cells are present there. Okay, so in just you remember, hematopoietic stem cells enters the medullary cavity, giving rise to the myeloid tissue. Okay, after that, primary center of the ossification is the diaphyseal center of the ossification. Okay, so this is another name of this primary center of ossification. Okay, so this primary ossification center is present in the diaphysis, so they are also called diaphyseal center of the ossification. So formation of the medullary cavity, and it is formed due to the osteoclast. Osteoclast, it just resorb the spongy bone, it just forms the medullary cavity in the center, and the hematopoietic tissue, hematopoietic stem cell go and give rise the enter into the medullary cavity and give rise their myeloid tissue. Okay, so now in this last step of the endochondral ossification, so there is a formation of the secondary ossification center. Okay, and we have primary ossification and the secondary ossification. Okay, so the main thing you should learn about this difference between these ossification centers is that, okay, so primary ossification center is present at the diaphysis, and the secondary is mostly present at the epiphysis. And also this primary center of ossification is before the birth, that is the main difference, but the secondary ossification center is shortly before or after the birth, okay, appears at one or both epiphysis. Okay, so this is very important difference between the primary and the secondary ossification. Okay, after that, periosteal bud. Okay, so this periosteal bud, as I have discussed, okay, so periosteal bud is forming the primary ossification center, but the difference in this secondary ossification center is that, okay, so the periosteal bud, it contains the mesenchymal cells and the blood vessel as it is. Okay, so it is similar like that of the primary center of the ossification. Okay, but in the secondary ossification center, we have this mesenchymal cell, blood vessel, and also this spongy bone, okay, in the interior retained, and no medullary cavity is there. Okay, so this is main difference between the secondary and the primary ossification center. Okay, so the thing I want to explain is that, so in the primary ossification center, it is formed by the periosteal bud. We everyone know it. Okay, so periosteal bud is the mesenchymal cell plus the blood vessel, but in the secondary ossification center, we have mesenchymal cell, blood vessels, and also the spongy bone, okay, is written at the interior. Okay, interior layer, interiorly it is retained there. Okay, but no medullary cavity is there. Okay, so those mesenchymal cell, blood vessels, and the spongy bone, combination of it forms the periosteal bud, and that is forming the secondary ossification center. Okay, so that is the concept. After that, after the completion of the secondary ossification center, there is a cartilage at the two places. Okay, so articular cartilage, epiphyseal cartilage. Okay, so after the completion of the secondary ossification center, so one is articular cartilage at the both articular in and epiphyseal plate. Okay, so epiphyseal plate is the junction at the epiphysis and diaphysis, and they are responsible for the growing the length. Okay, growing the length of the bone. Okay, so epiphyseal plate, so they are significant for that purpose. After that, union of the primary and the secondary ossification center is called the epiphyseal line. Okay, so this is also important. After that, long bones have one or two secondary ossification center, short bones have one ossification center. Okay, so you can learn this. Okay, so this is very easy to understand. So in this endochondral ossification, you just have to remember. Okay, so first of all, there is a formation of the cartilaginous model. Okay, and that cartilaginous model, okay, have the perichondrium, its outer layer. Okay, and in the first step only we have discussed, in that cartilaginous model there is a formation of the three zones. Okay, so first zone is a zone of proliferation, another is hypertrophy and maturation, and third is the provisional mineralization. Okay, so it is going in a sequence. First of all, there is division, after that there is a, okay, hypertrophy and the maturation, and after that there is a finally mineralization. So in this sequence there are the zones in the longitudinal section. Okay, in the longitudinal columns. Okay, and after that, when there is a proper formation, okay, of all those zone in the cartilaginous model, there is a formation of the bony collar. Okay, so bone, small Oban bone, immature bone like structure is deposited in the collar form. Okay, so bony collar is formed, and after the bony collar formation there is a perichondrium is referred as a periosteum, and with the formation of this bony column, and there is a formation of the, in the third step we have discussed about the periosteal bud. Okay, so mesenchymal cells and the blood vessel, they are commonly called periosteal bud. And after that we discuss with this medullary cavity formation, that is that is done by this osteoclast, and finally about the secondary ossification center. Okay, so in this pattern we have completed the all the steps of the secondary ossification center. Okay, so in your exam you just remember this point. Okay, formation of the secondary ossification center, and you just take the concept. Okay, as I have discussed here. So this concept, if you remember this in your exam, you can write in your own pattern. Okay, you can write in your own way. The main thing is the concept. If you get concept, you can write. If you do not have concept, and you just mug off all those things, okay, by heart all those things, then it will be difficult in exam. So they are the things of concept. Okay, so main thing is concept. If you take concept, then it will be very good. Okay, and our last topic is the difference between the Orban bone and the lamellar bone. Okay, and this is very easy, so I'll just recite all those points. Okay, so Orban bone, immature, they are mature. After that, collagen fibers are intertwined here, collagen fibers are orderly arranged here. More interfibrillar space is in Orban bone, and here less interfibrillar space is present. Hematoxyphilic matrix is there, and eosinophilic matrix is here. Okay, so when you do dust SNE stain, we can see the hematoxylin, okay, stain on the Orban bone, and eosino is stain on the lamellar bone. After that, formation and the mineralization is faster here, and it is slower here, and a lower mineral density, higher water content, higher mineral density, lower water content is in the lamellar bone. Okay, after that, matrix vesicle is the, okay, so by the matrix vesicles there is a mineralization, but here it is collagen mediated. Okay, in the lamellar bone. Okay, and after that, osteoclast can remove Orban bone totally, but here remove the portions of the lamellar bone at a time. Okay, so it can't remove totally. Okay, so there are the difference between the Orban bone. You can just learn it once, and you can write in the exam. Okay, so they are asked in a 1 marks or 2 marks question. Okay, sometimes. Okay, also they can ask in a viva. Okay, so Orban bone, lamellar bone, immature or the mature. Okay, so we have completed our topics for today's, okay, today's video. So we have discussed with the endochondral ossification, intramembranous ossification, and the difference between the Orban bone and the lamellar bone. Okay, so now in the next video we will be discussing further about other topics in this chapter, alveolar bone. Okay, and this much for today. We will meet in the next video, and I hope you are studying well. And for the notes of all those videos, you just can go to my Telegram channel. Okay, so the link of the Telegram channel I will be posting in the description below, so you can go into the Telegram channel and you can join there and take the notes. Okay, Okay, so we will meet in the next video. Till then, stay happy, stay healthy. Tata, bye bye. Thank you.